unfold.
Welcome, fellow explorers, to the deep dive. Today, we aren't launching a rocket toward the edge of the galaxy or decoding the scripts of a lost civilization.
No, not at all.
We are embarking on a far stranger, far more intimate odyssey one into our own inner space. We're taking a meticulous, highly detailed census of the most critical, yet least understood biological neighborhood within us. The human gastrointestinal tract.
Indeed. And this is really a deep dive into self-identity. We're confronting this idea that the human body, which we all perceive as this singular unified thing, is anything but.
Right.
The groundbreaking research we're looking at today lays out the concept of the holobiont.
A holobiont.
Yeah, and it's this powerful, maybe even unsettling idea that you are not simply an individual. You are a complex, dynamic, and integrated multi-species aggregate.
So we're essentially walking, breathing ecosystems.
Exactly, or what some researchers are now calling superorganisms.
A superorganism. That's the perfect framing for this. And our mission today is to move past that, traditional biology textbook definition of the gut.
Right, the passive tube.
Yeah, exactly. And instead, treat it not as this passive tube for our lunch, but as a genuine, highly complex second command center.
And this dive synthesizes just cutting-edge research across physiological homeostasis, neurological function, and even the most delicate aspects of mental health. And frankly, the implications they're revolutionary.
They absolutely are.
I mean, over the next half hour, the sources will correct a persistent, 50-year-old scientific myth about our own cells. They'll uncover the literal, dedicated hard wiring between the gut and the brain, a connection that operates at speeds we previously thought were impossible.
Wow.
And, maybe most profoundly, they provide overwhelming clinical evidence that the fate of your mind, from everyday stress to clinical depression, even to the acceleration of diseases like Alzheimer's, is being constantly negotiated by your microbial tenants.
It just changes everything we think we know about instinct and self-control. So, before we plunge into the detailed molecular biology and the trials, let's leave you, the listener, with the central premise of this entire journey. What if your deepest instincts, your most persistent cravings, and even significant aspects of your emotional destiny are being dictated by the trillions of occupants sharing your inner space, sending signals directly to your brainstem? Okay, let's unpack this. So we have to start where the popular conversation about the microbiome really began.
With that one big number.
Exactly. With a dramatic, almost cinematic statistic that dominated scientific discussion for half a century. I'm talking, of course, about the 10.1 myth.
Right. The belief that for every single human cell in your body, there were 10 microbial cells.
It made it sound like we were just passengers, overwhelmingly dominated biologically by our bacteria.
It was a very compelling narrative, and it captured the imagination for sure. It painted this picture of the human host as just a minor player in a game ruled by microbes.
But it was wrong.
Well, as our sources confirm, that figure was based on some extremely flawed and simplified methodology. The 10-to-1 ratio, it originated way back in 1972 from a simple, informal calculation.
Like a back-of-the-envelope guess, not a rigorous census.
Precisely. It's what scientists call a Fermi problem estimation, by a microbiologist named Thomas Luckey.
And what was the fundamental mistake he made?
The mistake was an assumption of uniformity that just doesn't exist in the human body. Luckey assumed a constant high bacterial density of 10 to the 11 bacteria per gram across the entire GI tract.
The whole thing, from stomach to colon.
The whole thing. He treated the highly acidic, fast-moving, sparsely populated stomach and small intestine the same as the high-density, slower colon, like it was all one uniform fermentation tank.
Which of course it isn't. That makes perfect sense. The upper GI tract is a hostile environment. It's designed to kill bacteria and move food through quickly.
Exactly. You can't have the same population density there. So this is why modern rigorous analyses were so necessary. The definitive correction came with a 2016 study.
The one from Sender, Fuchs, and Milo.
That's the one. They provided the revised reality. Using modern sequencing, cell staining, and crucially, accounting for that huge variation in densities, they got a much more accurate count.
So what did their new count reveal? What's the real number?
They estimated the total number of bacterial cells in an average reference man at approximately 3.8 times 10 to the 13th.
Which is still a staggering number.
Oh, absolutely. But crucially, they pair this with a similarly rigorous update of the total number of human cells, which they estimated at about 3.0 times 10 to the 13th.
So the numbers are much, much closer. The dramatic 10.1 ratio just evaporates.
It's gone completely. The new ratio is far more egalitarian. It's about 1.3.1.
1.3 to 1.
Yep. We are not vastly outnumbered. We are in a very tight, near one-to-one partnership with our microbial tenants.
Okay, so if the numbers are so much closer, does that mean we should worry less about the microbiome? Has the urgency been, downgraded?
That's a crucial question, and the answer is an emphatic no. The microbial mass may be smaller, around 200 grams, not a couple of kilograms. And the numerical superiority is downgraded, but the functional superiority, that remains absolutely overwhelming. This is the critical takeaway. Quantity is secondary to capability.
The genetic reservoir, that's where the real power of this partnership lies, isn't it?
Precisely. I mean, consider your human genome, your blueprint. It contains roughly, 20 to 25,000 protein-coding genes?
Correct.
That's the whole instruction manual for building you?
Right.
But when you factor in the aggregate genome of your microbiome, the metagenome, you get access to millions of unique genes.
Millions?
Yes. We are talking about a second genome that dramatically expands your metabolic repertoire by a factor of 100 or more. Your human genome is like owning a smartphone. Great for core functions.
But the microbial genome is the entire internet.
That's it, exactly. You've outsourced the heavy lifting of survival to them.
Can you give us a tangible example of a function we gain from that?
Absolutely. The human genome is great at building things and regulating hormones, but we just lack the enzymes to break down many complex plant molecules, specifically polysaccharides fibers like xylan, pectin, resistant starches.
The MACs, microbiota accessible carbohydrates.
The very same. If we eat them, they pass right through us. But the microbial genome has the specialized genes to ferment these MACs, extracting massive amounts of energy and, critically, producing the signaling molecules we rely on.
And beyond energy, what else is in that microbial library?
Well, they're essential for synthesizing key vitamins we can't produce ourselves. Best examples are vitamin K for blood clotting and B group vitamins, especially B12, which is critical for nerve health.
And they protect us too, right?
Yes, they're experts at metabolizing xenobiotics compounds that are foreign to the body, including many drugs and environmental toxins. We rely on them for that metabolic shield.
That genetic power makes this next section, the diversity crisis, profoundly alarming. If the microbiome is our metabolic operating system, it sounds like the modern Western world is. Well, it's undergoing a mass extinction event inside us.
That is the tragic reality. When researchers compare the microbiome of industrialized Western populations with, say, the Hadza people of Tanzania.
The hunter-gatherers.
Yes, the difference is stark. It's like comparing a monoculture field to a tropical rainforest. The Hadza, who live a lifestyle very close to our Paleolithic ancestors, have a gut ecosystem that is just vastly richer and more diverse than ours.
So what kind of ancestral symbionts are they harboring that we in the West are losing?
Well, they host robust communities of bacteria that are rare or entirely absent in Westerners. The sources point to a fascinating example. Treponema succinifacans.
Treponema, isn't that genus associated with?
With pathology, yes. Syphilis, specifically. But in the Hadza gut, it appears to function as a crucial partner, specializing in breaking down the complex carbohydrates they consume from their environment.
So we've lost species that were once beneficial just because we changed our diet and our environment.
Precisely. Our modern lifestyle hygiene, processed low-fiber diets, pervasive antibiotic use, has essentially sanitized and starved the ecosystem. The sources suggest this chronic, low-grade industrialized dysbiosis is strongly correlated with the rise of the diseases of civilization.
Like asthma, allergies, metabolic syndrome.
All of it. We've inadvertently stripped away our ancestral defense layer.
You mentioned an anomaly with bifidobacterium, a microbe we usually think of as healthy. We buy it in probiotics.
This detail really makes you question what a healthy microbiome even is. Bifidobacterium is ubiquitous in Western guts, often because of dairy and supplements.
But not in the Hadza.
In adult Hadza people, this ubiquitous genus is conspicuously absent.
Wow, that is counterintuitive. Does that mean high levels of bifido in our gut might be a signature of industrialization, not ancestral health?
That's the implication. It challenges the assumption that a healthy state looks the same for everyone. What we call normal, based on sampling us, might actually be a marker of a degraded ecosystem compared to the ancestral state. Context is everything.
And the Hadza data also revealed something unique about their social structure, reflected in their microbes.
It's a fantastic nuance. Researchers observed a significant sexual dimorphism in the Hadza microbiome, linked directly to their division of labor.
The men hunt, the women forage.
Exactly. The men, who focus on game and honey, have distinct microbial profiles compared to the women, who primarily forage for tubers and berries.
Different diets creating different internal gardens.
Precisely. The stratification of what they eat creates different ecosystems. It's a dynamic richness that's largely lost in our homogenized, supermarket-driven diets, where the difference between a male and female diet might be, marginal at best.
So we've established we're a 1.1 ecosystem, but the overwhelming computational power resides with the microbes. Now, let's get into how this ecosystem talks to the brain, not through slow signals, but through literal, dedicated hardwiring. We have to start with the enteric nervous system, ENS, the second brain.
The ENS is one of the marvels of anatomy, often overlooked because it's buried deep within the gut wall. It's this sprawling, intricate network of neurons.
And what's the scale we're talking about here? How vast is it?
It's astonishing. The ENS contains approximately 500 million neurons.
500 million.
Yes. To put that in context for you, that is 5 times the neuron count of your entire spinal cord. And it's about 2/3 the total number of neurons in a cat's entire nervous system.
So this is not a simple relay station. It's a massive independent computational hub.
Completely. And it's highly organized. It's composed of 2 dense nerve networks, or plexuses. First, you have the myenteric plexus, or Auerbach's plexus. It's situated between the muscle layers of the gut wall. This is the gut's motor drive. It controls peristalsis, the rhythmic movements that push everything along.
The engine room.
Exactly. Then you have the submucosal plexus, or Meissner's plexus, which is closer to the inner lining. This is the chemical reconnaissance unit. It regulates blood flow, secretion, and most critically, senses the chemical environment of the gut.
And its autonomy is the key thing.
Absolutely. The sources stress that the ENS can coordinate the entire complex process of digestion, secretion, motility. All of it, perfectly, even if the vagus nerve connecting it to the brain were completely severed.
It doesn't need permission from the head to operate.
It has its own processing capability. That's why we call it the second brain.
Okay, here's where it gets really interesting. For decades, the thinking was that the gut communicated with the brain slowly.
Right, like surface mail. Hormones in the bloodstream.
Exactly. Minutes later, the brain gets the signal. But that whole paradigm was shattered.
It was shattered by the discovery of the neuropod cells by Dr. 𝐏𝐎𝐃𝐂𝐀𝐒𝐓 and his team. This discovery changed the timeline of gut-brain communication from minutes to milliseconds.
So tell us about the mechanism. What makes these cells so revolutionary?
Neuropod cells are specialized enteroendocrine cells. We used to think they just released hormones into the blood. But 𝐏𝐿𝐴 has discovered they have this unique projection, like a foot or neuropod.
A foot?
Yeah, and this foot extends right through the gut wall to form a direct bona fide synapse with vagal neurons.
A direct synapse. So it's not chemical mail. It is structurally and functionally an optical fiber cable.
That analogy is perfect because it explains the speed. It's a direct synaptic connection, so it bypasses the circulatory system. These cells use fast neurotransmitters, specifically glutamate, which means signals transmit in milliseconds.
Which is the neurobiological basis for what we call a gut feeling.
That's it. That immediate visceral communication that bypasses our slower analytical brain.
And the sources cite this brilliant experiment that demonstrates the specificity of this communication, something with sugar versus sweeteners.
Yes, it's an elegant study. It shows the gut has its own sense of taste for nutrients. In the experiment, when neuropod cells were exposed to caloric sugar, sucrose, they immediately released glutamate at the synapse.
Okay.
This specific glutamate signal instantly triggered a distinct vagal pathway, signaling nutrition and reward straight to the brainstem.
But what about the non-caloric sweeteners, like sucralose?
Well, the cells were activated, they detected the sweetness, but they triggered a different neurotransmitter pattern involving ATP. And crucially, that ATP-based signal failed to satisfy the brain's deep reward centers.
So the brain gets the message sweet, but not the message energy.
Exactly. It gets something sweet is here, but it fails to receive the vital reinforcing message that says, this is valuable caloric energy.
And this has profound implications for cravings and appetite.
It suggests our subconscious drive for energy is being regulated by these direct, instantaneous signals. If you're constantly feeding your system non-caloric sweeteners, you satisfy the tongue, but the neuropods fail to send the true reward signal, leaving you perpetually unsatisfied and driving that constant search for energy.
So the microbes, through this pathway, are literally lobbying the brain for resources.
In milliseconds.
So we've mapped the hardwiring, now we shift to the molecular output, the chemical factories. And we have to start with the great paradox of a famous mood molecule, serotonin.
Right. When we hear serotonin, we think brain, we think mood.
But that's not the whole story.
Not even close. The paradox is that the brain supply of serotonin is a tiny fraction of the total. About 95% of the body's entire stock is manufactured, stored, and used almost exclusively within the GI tract.
So the gut is the primary production center. Where does it all come from?
It's produced by enterochromaffin, EC cells, which are specialized cells in the intestinal lining. They synthesize it from the amino acid tryptophan using a specific enzyme, TPH1.
And the distinction between the gut's TPH1 and the brain's enzyme is critical here.
It is, because the two pools are separated. The peripheral serotonin produced by TPH1 in the gut cannot cross the blood-brain barrier in a healthy adult.
So this massive supply of gut serotonin isn't directly regulating your conscious mood.
Correct. But the two systems are linked, and the gut needs this chemical workhorse for its own functions.
Yeah.
And this is where the microbiome takes control. They're the master switch.
How so?
Spore-forming bacteria, particularly Clostridia, and their metabolic byproducts, which we'll get to, have been shown to upregulate the expression of that TPH1 enzyme, dramatically boosting serotonin synthesis.
So if you remove the bacteria, what happens to the serotonin supply?
It plummets. Germ-free mouse models show significantly reduced levels of serotonin, and this deficit can be completely reversed just by colonizing the gut with specific strains, like Lactobacillus ruminus. It's direct proof.
So if it's not for mood, what is all that serotonin doing in the gut?
It's a vital, multifunctional workhorse. It regulates peristalsis by stimulating the myenteric plexus. It regulates secretion of fluids. And critically, it modulates visceral sensation and pain perception.
The intensity with which we feel discomfort in our abdomen.
Precisely, which immediately connects to common disorders.
Like IBS.
Exactly. When serotonin release is excessive or uncontrolled, it's directly associated with the symptoms of IBS diarrhea, hypersensitivity. Too little leads to constipation. The microbes maintain that delicate equilibrium.
Okay, from the workhorse of serotonin, let's go to the actual chemical currency of the entire axis. Short-chain fatty acids, or SCFAs.
If the gut is a chemical factory, SCFAs are its most critical product. They are small organic compounds, primarily acetate, propionate, and butyrate produced when bacteria ferment those non-digestible fibers, the MAs.
And without fiber, the factory shuts down.
It does. And we should really break down that metabolic triad because each SCFA has a distinct specialized role.
Let's start with butyrate.
We have to. Butyrate is perhaps the most critical for local gut health. It is the preferred energy source for the colonocytes. The cells lining the colon meeting up to 7% of their energy needs.
It's literally food for the gut lining.
It is. And its primary function is to maintain the integrity of that lining. It strengthens the tight junctions, the seals between the cells. When those weaken, the gut becomes leaky. Butyrate repairs those seals.
And what about its role in inflammation?
It's intensely anti-inflammatory. It does this by inhibiting enzymes called histone deacetylases, or HDACs. By inhibiting HDACs, butyrate changes gene expression in immune cells, keeping the immune system balanced and preventing overreactions.
A healthy, sealed, peaceful colon requires butyrate.
Yes. Next is propionate. It's absorbed into the portal vein and travels to the liver, where it helps regulate gluconeogenesis, glucose production, and satiety. It's the gut telling the host, I've been fed.
And we'll come back to its neuroprotective role later.
We will. And finally, acetate. It's the most abundant. It enters the systemic circulation, and unlike gut serotonin, it can cross the blood-brain barrier.
But what does it do in the brain?
It influences hypothalamic function, playing roles in appetite, energy expenditure, and metabolism.
It sounds like our entire metabolic health, from local gut integrity to systemic energy and inflammation, rests on the efficient production of these three molecules.
That's absolutely correct. And that efficiency relies on a highly sophisticated and fragile structure we call ecosystem interdependence.
Cross-feeding.
Cross-feeding. This is where the loss of one single species can collapse the entire process.
Walk us through an example of that.
Certainly. Some key primary fermenters, like certain strains of bifidobacterium, produce acetate and lactate, but they don't make butyrate.
Okay.
Other secondary bacteria, like the crucial anti-inflammatory species Faecalibacterium prausnitzii, can't break down the fibers themselves. Instead, they use the acetate and lactate from the primary fermenters as raw material to create the vital butyrate.
So if you lose the bifidobacterium link due to antibiotics or diet...
You don't just lose acetate. The critical butyrate-producing bacteria, F. prosnitzi, essentially starve because their food source is gone. This leads to a catastrophic deficiency in butyrate, which compromises the gut barrier, increases inflammation, and starves the colonocytes.
It shows how quickly dysbiosis can lead to deep metabolic failure.
Exactly.
We've established the ENS as the second brain and SCFAs as the currency. Now we tie it all together with the ultimate piece of anatomical hardware, the vagus nerve.
Cranial nerve X, the information superhighway.
But the traffic pattern on this superhighway is, well, it's pretty counterintuitive if you think of the brain as the commander.
It is, because the traffic is heavily biased in one direction. The sources highlight that a vast majority, an estimated 80% of all vagal fibers are first.
80%?
80%. That means they are sensory fibers, carrying detailed information from the gut to the brain. The downward efferent traffic, which carries commands, is only about 20%.
So the brain is dedicating 4/5 of its bandwidth just to listening to the gut. What kind of status reports are coming through this highway?
Highly detailed, continuous sensory reports. The vagus nerve endings are sensing everything. The chemical composition of the lumen, the degree of physical distension, the presence of inflammation, the concentration of microbial metabolites.
All of it.
All of it. And this continuous influx of data constantly shapes our emotional state and stress response, often entirely below the level of conscious awareness.
That 80% afferent bias fundamentally flips the script. It suggests our gut is the primary informant.
It forces us to reconsider the origin of some emotions. That sinking feeling in your stomach. It may not be a psychological reaction to a thought. It may be a direct ascending sensory input about a negative physical state, say mild inflammation, that the brain then interprets as anxiety.
The feeling originates in the body before the cognition assigns it a name.
Exactly. Now let's look at that critical 20% of traffic moving down the highway.
The efferent signals, the brain's commands.
Right. And these aren't just about movement. They are about control, particularly the immune system.
Yeah.
This is the inflammatory reflex. It's the brain's dedicated emergency brake on runaway immune overreaction.
How does that brake system work at the molecular level?
When the brain detects systemic inflammation, either through those afferent signals or circulating danger signals, it initiates a precise counter-response. It sends inhibitory signals down the vagus, which cause the release of acetylcholine near the gut wall.
And what does that acetylcholine do to the immune cells?
It interacts with specialized immune receptors, alpha-7 nicotinic receptors, on local immune cells like macrophages. This interaction instantly inhibits the release of highly destructive pro-inflammatory cytokines.
So it tells the immune system, stand down.
In essence, yes.
Yeah.
We have enough information. The brain is actively managing the immune status of the gut.
And if that brake fails?
Well, dysregulation of this pathway, a failing brake, is often characterized by low vagal tone. And that is a major hallmark of chronic inflammatory conditions, particularly IBD and inflammatory subtypes of IBS. If the brain can't push the brake hard enough, inflammation persists.
Okay, so the vagus highway ensures the gut state is constantly influencing our physiology. But the most disruptive research connects gut microbes directly to our most complex emotional states.
Moving from correlation to direct causation.
Right. We are talking about studies that literally demonstrated you could be transferring the blues.
This was the moment the field truly moved into clinical credibility. For years, the idea was dismissed as too speculative. But studies, like Kelly et al. In 2016, provide definitive causal evidence.
So walk us through the design of that study. It has huge philosophical implications.
It was a classic fecal microbiota transplantation, or FMT, experiment. Researchers took fecal samples from human patients with active major depressive disorder and transplanted them into microbiota-depleted rats. The control group got samples from healthy subjects.
And the staggering result was that the recipient rats adopted the emotional state of the human donor.
Exactly. The rats that received the depressive ecosystem exhibited clear behavioral phenotypes of depression. They showed signs of anhedonia, the inability to feel pleasure, by showing a reduced preference for sugar water.
Wow.
They also showed dramatically increased immobility in the forced swim test, a reliable proxy for behavioral despair.
So the depressive state was literally transmissible. What was the transferred ecosystem doing chemically to cause that?
It was a complex chemical assault. The depressive ecosystem disrupted the host's use of tryptophan.
The precursor to serotonin.
Exactly. But in the depressed state, the microbes diverted tryptophan away from the beneficial serotonin pathway and shunted it towards the neurotoxic kynurenine pathway.
The kynurenine pathway, how does it cause neurotoxicity?
When tryptophan is shunted into this pathway, it creates a series of metabolites, including, critically, quinolinic acid. Quinolinic acid is a known excitotoxin. It overstimulates neurons and contributes to inflammation, potentially crossing the blood-brain barrier to trigger neurotoxic effects in the brain.
So the gut microbiome essentially activated a metabolic shunt that generated inflammatory toxin.
And created the depressive state in the new host.
Yes. And that deep realization has led directly to this exciting new therapeutic field of psychobiotics.
Psychobiotics. They're defined as live organisms that, when ingested, confer a mental health benefit. They are designer microbes developed to modulate the gut-brain axis.
And the sources pinpoint specific strains that have gone through rigorous trials.
The most well-documented combination is Lactobacillus helveticus R0052 and Bifidobacterium longum R0175. These strains don't just aid digestion, they actively engage and calm the host's stress response.
What are their observed therapeutic mechanisms?
Yeah. They're powerful modulators of the HPA axis, the body's stress pathway. They're shown to reduce circulating levels of the stress hormone cortisol. And, crucially, they increase the expression of brain-derived neurotrophic factor, or BDNF, in the hippocampus.
BDNF is often called miracle grow for the brain.
Exactly. It's essential for creating new neurons and enhancing synaptic plasticity. By reducing cortisol and increasing BDNF, these strains foster a healthier, more resilient brain structure.
And the clinical evidence is compelling.
Extremely. RCTs show this formulation significantly reduced depression and anxiety scores. One study in cancer patients reported a remarkable 60% improvement in depression status after just 1 month.
And we have to circle back to the Vagus highway here. It's essential for this effect, isn't it?
Absolutely vital. The Lactobacillus rhamnosus JB1 study is the hard proof. This strain reduced anxiety in mice. But, and this is the key finding, when the vagus nerve was surgically cut, those anxiety-reducing effects were completely abolished.
So the bacteria need the hardline connection to send their message of calm.
Correct. The benefit is dependent on the neural connection.
Now let's pivot from mental health to neurodegenerative destiny, the Alzheimer's connection. For decades, the view of Alzheimer's was so simple, just a plaque problem in the brain.
That neurocentric view is crumbling, Homer. The emerging perspective suggests the amyloid beta plaques may be initiated or drastically accelerated by chronic inflammation originating in the gut.
This takes us to the molecular mimicry hypothesis. What exactly is being mimicked?
It's fascinating. Certain gut bacteria, like Escherichia and Salmonella, produce protein structures called amyloid fibers, specifically a type known as 𝐏𝐿. These bacterial amyloids are structurally almost identical to the AB plaques that characterize AD.
So the bacteria are practicing molecular camouflage.
Essentially, the hypothesis is that this mimicry triggers a chronic immune response in the gut. The body, recognizing the bacterial amyloid, may accidentally produce antibodies or endogenous amyloid that eventually cross-seeds the pathology in the brain.
And this is all made worse by the dysbiosis seen in AD patients.
Precisely. Studies show a consistent pattern in AD patients. A decrease in beneficial anti-inflammatory species and an increase in pro-inflammatory ones. This compromises the intestinal barrier, leading to leaky gut.
And when the gut leaks, toxins get into the bloodstream.
And that's where lipopolysaccharides, or LPS, come in. They're potent endotoxins. Once they leak into the blood, they cause systemic inflammation, which activates the microglia, the brain's immune cells. Activated microglia release inflammatory cytokines, which accelerate plaque formation and drive neuronal death.
So the gut is literally fueling the brain's decline.
Is fueling the fire. But the sources end this section on a remarkable note of hope, involving one of those SCFAs, the propionate defense. This is very recent.
From 2025 research?
Yes. The study showed that propionate, the SCFA produced by bacteria like Akkermansia mucinifera, had significant efficacy in mouse models of AD.
And how did it achieve that?
It significantly reduced the amyloid plaque burden and dramatically dampened astrocyte reactivity, the overactivation of supporting brain cells that causes inflammation. It seems to work by dampening the effect of the inflammatory cytokine IL-17.
So future Alzheimer's treatments might not just be neurological drugs, but metabolic therapies targeting the gut.
The path to the aging brain may quite literally run through the metabolic health of the gut.
We've seen the immense power the microbiome wields. Now we need to discuss where this inner world begins and how quickly it can be ruined. The genesis is the seeding process at birth, specifically C-section versus vaginal birth.
The mode of delivery is the absolute defining event for the infant's initial ecosystem. A vaginally born infant is immediately inoculated with the mother's vaginal and fecal microbiota lactobacillus bacteroides. It's the critical priming event for the immune system.
And the C-section infant misses this microbial baptism entirely.
They do. They bypass the birth canal and are instead colonized by skin flora like Staphylococcus and hospital microbes. This results in delayed colonization by beneficial species and lower initial diversity.
And this isn't just a transient difference, it has long-term health consequences.
Significant ones. The sources cite the extensive 𝐏𝐿-𝐃 study, which showed that this C-section signature at 1 year of age correlates with a markedly increased risk of developing childhood asthma and other allergic diseases.
The mechanism is essentially a failure of proper immune calibration.
That's the leading hypothesis. The lack of that maternal microbial legacy disrupts the development of T-regulatory cells, which teach the immune system what not to react to. The immune system is improperly calibrated.
But the study also offered some hope that this isn't a life sentence.
Exactly. They found a crucial window of opportunity. If the microbiome of a C-section baby normalizes within that first year through breastfeeding or probiotics, the increased asthma risk recedes back to baseline.
So the failure of immune maturation can be mitigated if the inner garden is restored quickly.
It can be. Now, from Genesis, we jump to the most damaging event for the adult microbiome. The apocalypse brought on by antibiotics.
Oh, wildfire.
The scale is hard to grasp. A single course of broad-spectrum antibiotics can reduce the microbial load by 99.999% in hours.
That's total devastation.
It is. While the total number of bacteria may recover, the diversity is often permanently scarred. Specific ancestral species may go locally extinct within the host, never to return without reintroduction.
And antibiotics don't just reduce the head count, they change the physical environment.
They induce a profound chemical shift. They kill off the obligate anaerobes, the oxygen-consuming good guys. This leads to a massive increase in the gut's oxygen levels, raising the redox potential.
And that increased oxygen favors the invaders.
Precisely. That oxygenated environment is no longer suitable for the beneficial anaerobes. Instead, it creates the perfect breeding ground for pathogens, like the notoriously problematic Clostridioides difficile. It creates a dysbiotic loop.
That environmental shift brings us to the final chapter, restoration. The need to restore this community is what validated a very ancient and frankly kind of gross medical practice.
We are, of course, talking about 𝐏𝐿-𝐃𝐂𝐀𝐒𝐓 yellow soup from 4th century China.
Huanglong Tang.
Yes. 𝐏𝐿, a brilliant physician, prescribed this preparation for severe life-threatening diarrhea.
And that preparation was effectively the first documented fecal microbiota transplantation, or FMT.
It was. He prescribed a suspension of fecal matter from a healthy person. He didn't have the language of bacteria or redox potential, but his clinical observation was infallible. He knew he was reseeding the sick gut to outcompete the pathogen.
And centuries later, that ancient wisdom became necessary to solve the modern crisis of antibiotic-resistant C. difficile.
A crisis of medicine failing itself. Standard antibiotics were making the problem worse. This crisis necessitated the modern resurrection of FMT, culminating in the FDA approval of VOWST in April 2023.
VOWST is essentially the pharmaceutical-grade, sanitized evolution of yellow soup.
It is the first FDA-approved, orally administered fecal microbiota product. It uses highly purified, dormant spores of Firmicutes bacteria derived from rigorously screened donors. The key is the spore form, which can survive stomach acid.
And what was the efficacy in the trials?
The phase 3 trials showed dramatic results. VOWST reduced the recurrence of C. diff infection to just 12.4%, compared to nearly 40% in the placebo group. It's a revolutionary success.
And the mechanism reinforces everything we've talked about.
Exactly. The spores germinate in the colon, they outcompete C. diff for resources, and, more specifically, they restore the crucial conversion of primary bile acids to secondary bile acids. Secondary bile acids are highly antagonistic to C. diff growth. The ecosystem heals itself.
So the approval of VOWST really marks a pivotal moment, transforming this into a standardized, approved medicine, #tag out UTRO, the holobiont horizon. This deep dive into the inner cosmos really reveals that the self is not a singular entity, but a profound, complicated negotiation.
Between multiple species,
Yeah. The sources confirm that we are holobionts, engaged in a constant dialogue with our microbial tenants.
The evidence is just exhaustive. Demographically, we are a powerful 1.1 partnership, but genetically we are completely dominated by the microbial side.
Neurologically, they're hardwired to us via the vagus nerve and those millisecond-fast neuropod cells.
Constantly influencing our desires and emotional state. And chemically, our health depends on them for 95% of our serotonin and all those vital SCFAs that fuel our colon, regulate our metabolism, and manage inflammation.
This journey forces a fundamental rethinking of medical philosophy, moving away from that 20th century mindset of war on germs.
That paradigm is being rapidly replaced by the stewardship of a garden. We now recognize that the health of the second brain is just inextricably linked to the destiny of the first.
Whether it's depression, inflammatory diseases, or Alzheimer's, the path to treatment increasingly targets the metabolic powerhouse in our gut.
We are the architects and caretakers of our own inner space.
And that brings us to our final provocative thought for you, the learner. If our deepest cravings, our innate instincts, and even the feeling of anxiety are shaped by instantaneous, unconscious signals sent via the vagus nerve signals that dictate what we pursue and how we feel, how much of what we call me is truly our own autonomous identity?
And how much is merely a reflection of the constant, necessary microbial negotiation we carry within?
That is the ultimate question of the Holobion, and one we're just beginning to answer. Until next time, keep digging.